Pose with the fans. Pose with the pose with the pose with the fans. Pose with the pose with the pose with the fans. Pose with the fans. Pose with the fans.
Imagine if every cell in your body suddenly didn't have enough energy. When your brain starts to fail, your muscles weaken, and even breathing becomes a struggle. That is the reality for patients with Leigh syndrome.
What is Leigh syndrome?
Leigh syndrome is a rare genetic disorder that caused by mutation in mitochondrial DNA or nuclear genes that disrupt the cell's main energy producing system, which is electron transport chain or ETC complexes, especially complex 1, complex 4, or ATP synthase. This condition leads to impaired ATP production and lactic acidosis.
Let me show you how. This process occurs across the inner membrane of the mitochondria. The matrix is towards the bottom and the intermembrane space is towards the top. NADH and FADH2 has been reduced with electrons and protons by previous reaction in the Krebs cycle. This process is the last step of metabolism, resulting in the production of water and ATP. NADA reduces complex 1 with 2 protons and 2 electrons. During this process, 2 protons are shuttled from the matrix into the intermembrane space. Ubiquinone carries the protons and electrons to complex 3, where the protons are moved to the intermembrane space. FADH2 reduces complex 2 with 2 protons and 2 electrons. Ubiquinone carries these protons and electrons to complex 3, where these protons are also moved to the intermembrane space. These 4 electrons are transported one by one to complex 4 by cytochrome C. For each electron received by complex 4, a proton is shuttled from the matrix to the intermembrane space. The 4 electrons then combine with 4 protons in the matrix and oxygen to make water. The proton gradient established throughout this process is used to make ATP. ATP synthase takes protons from the intermembrane space, rotates and pumps them into the matrix. For these 3 protons pump, an ATP is made from ADP and inorganic phosphate. The rotation of the protein drives this catalytic reaction. But in the ETC of someone with Leigh syndrome,
let me show you how. If complex 1 is mutated, some electrons leak to oxygen forming superoxide, a reactive oxygen species. Then, complex 2 also sends electrons to coenzyme Q but doesn't pump protons. Next, complex 3 passes electrons to cytochrome C. When flow is disrupted, it leaks more electrons, creating extra ROS. At complex 4, electrons combine with oxygen to form water, but if it's mutated, electrons back up, boosting ROS at complex 1 and 3. Finally, at ATP synthase, protons from the intermembrane space rotate through ATP synthase back into the matrix, producing ATP. Too much ROS damages mitochondria and cuts energy output. Since energy production is disrupted, tissues that require large quantity of energy, such as brain and muscle tissues, are the first to suffer.
Developmental retardation and muscle weakness are the symptoms of Leigh syndrome. It is hard for them to walk, talk, or play like other children. Not only that, respiratory failure is also a symptom of Leigh syndrome that causes patients to have difficulty breathing. Finally, brain lesions are visible on MRI.
Unfortunately, there's no cure for Leigh syndrome, but there are some treatment options that can help manage the condition. These include the consumption of coenzyme Q10 or riboflavin supplements to help support the ETC function. Second one, we can also practice ketogenic diet to reduce the reliance on glucose metabolism. And in the future, maybe gene therapy, which is still under research.